sulcus, which is this inferior ramus here, that is the calcarine sulcus. And where the stem of the Y meets, this is the parieto-occipital sulcus. So we trace the parieto-occipital sulcus. So here you can see this just falls short in the suprolateral surface. So here you can see this is the parieto-occipital sulcus which we have traced. Everybody has seen? Everybody should be knowing, right? You can see this. What is this sulcus? That's the calcarine sulcus. And what is this? That's the parieto-occipital sulcus. What's this? And this is the. So here you can see we've traced this parieto-occipital sulcus along the suprolateral surface. So we have an imaginary line which joins this parieto-occipital sulcus with the pre-occipital notch. So this would be the area. See, this notch is there. This would be the area of the pre-occipital notch in this specimen. So we join the parieto-occipital sulcus with the pre-occipital notch. An imaginary line joining the 2 points. Which 2 points? Tell me. Pre-occipital notch. This imaginary line, the area below this line will form the occipital lobe. The area behind this, below and behind this, will form the occipital lobe. Then what do? We had the central sulcus. We extend this posterior ramus of the lateral sulcus to meet this imaginary line which we just drew by joining the pre-occipital notch and the parieto-occipital sulcus. So when we extend this imaginary line, what will happen? We demarcate an area placed just below or inferior to the posterior ramus of the lateral sulcus. So this area forms the temporal lobe, and the area between the central sulcus and the imaginary line here and above the posterior ramus of the lateral sulcus will form the parietal lobe. So that's how we demarcate the parietal lobe. Alright? All the 4 lobes. Now, after the lobes, what are the borders and surfaces of the cerebral hemispheres? Right? We have 3 surfaces, namely, you can see this surface. This is the medial surface, and you can see it's almost vertical in orientation. Can you see? It's almost vertical. See? Right? This is the medial surface. Then we have another surface, the broadest of all. This is the superolateral surface. This is the superolateral surface. Then we have this surface. This is the inferior surface. This inferior surface is further subdivided into 2 subdivisions. Anteriorly placed, this is the orbital part of the inferior surface. This is the orbital part. Why do we call this as the orbital part? This is a part of which lobe? This is a part of the frontal lobe, and this is the inferior surface or the orbital surface of the frontal lobe. Now, if I place it against the cranial cavity, this surface will rest against which cranial fossa? Anterior cranial fossa. Which plate? The orbital plate of the anterior cranial fossa, orbital plate of the frontal bone. You remember the area just lateral to the cribriform, right? Cribriform fossa. So this area, that's why it's forming the roof of the orbit. So that's why this is known as the orbital surface. And then this posterior 2/3, rest of this area forms the tentorial part. Tentorial part of the inferior surface. Why? Because what rests here? What intervenes in between this area? That's the tentorium cerebelli. Separates this surface from the surface of the 2 cerebellar hemispheres. So that's why this surface is known as the tentorial surface. Okay? Now the borders. We have a medial border, right? This is the medial border. Then we have an inferomedial and a infrolateral border. Infromedial and a infrolateral border. And here you can see this border, infrolateral border here. This portion of the infrolateral border is also known as the superciliary border. Okay. So we divided it into borders and surfaces. Now here you can see what is placed outside, and in this cut section also you can see here, look at this thin rim of the grey matter. Can you differentiate the grey matter from the centrally placed white matter? Look at this. In this section, this is the cortex, right? This is the cerebral cortex. Can you see the grey matter there?
Yeah.
Right? So that's the cerebral cortex, and it's that's a very thin rim of the cortex, and placed in the center of the cortex is the white matter, right? You can see this is the white matter of the cerebral cortex. Now you can see certain indentations and elevations there, right, on the surface. Now during development, can you tell me the three parts of the neural tube which gave rise to the brain, different parts of the brain? Neural tube was divided into two tubes, prosencephalon, mesencephalon and the rhombencephalon. So this area, what you see here, the cerebral cortex, basically this area, basically developed from the prosencephalon. And the two subdivisions of the prosencephalon are the telencephalon and the diencephalon. So the telencephalon form the cerebral cortex, whereas the diencephalon give rise to the thalamus and the hypothalamus. So what happened? This telencephalon overgrew in size the diencephalon. What happened? The diencephalon's growth was very small, so it became sandwiched between the inferior and the middle of the prosencephalon, so the telencephalon. So, and in this telencephalon also, the growth was more in the region of the cortex. The cortex was growing rapidly, right? So the cortex has how many layers? Here in this, it has, that's the old cortex which has 3 layers, the hippocampal and the piriform, whereas the new cortex, it has 6 layers, right? So there was a lot of growth occurring in the region of the cerebral cortex. But this growth, due to this growth, but there was a limited area in the cranium. The cranium cavity was limited. So to accommodate this increased growth of the cortex, what happened? There were certain involutions in the cerebral cortex, and due to these involutions, what happened? Certain areas were raised and the certain areas became depressed, and we named these raised areas as the gyri and the depressions as the sulci. Alright. Now, these gyri and sulci are not constant except the major gyri and sulci, like the central sulcus, right, and some of the major gyri, but they differ in each individual. Alright. So, to make our study easy, we have given names to these gyri and sulci. Okay. Now let us see what are these important gyri and sulci. First I will draw and then we'll trace it on the surface of the cerebrum.